Chapter 2, Section 8, Endospores. Now, endospores are a specialized type of spore found in the microbial world. Okay, these are differentiated cells that are metabolically dormant. Because they're metabolically dormant and their composition, we find them to be resistant to heat, radiation, chemical exposure, drying, even the lack of nutrients has no effect on these structures because there's no metabolism. Everything is set in almost a gel/crystalline state, proteinaceous crystal states. This is a survival mechanism. Okay, bacteria use this when the nutrients are all gone in a certain area, or water starts to dry out, go away, evaporate. This is a survival mechanism. So yes, the majority of the cells present will probably die before they can form these spores, but enough of that population will survive that the species will survive for when the water comes back, the nutrients come back. What we see happens is a vegetative, metabolically active cell gets some some extracellular stimulus. Something happens, something changes in the environment, and then they will form these endospores, these metabolically inactive things. And they can stay that way for days, weeks, months, years, decades. And then they can return to a vegetative, metabolically active state. When the bacteria turn into endospores, this is great for dispersal. Wind, water, maybe whatever they're in, that spore happens to be in, gets taken up when an animal is eating. A herbivore is eating plants. Well, the dust on that plant leaf is going to be some endospores. The gut, the ruminant gut of that cow, that goat's not going to matter. Not going to do anything to that spore. They'll pass out in the feces somewhere else. When we stain bacterial smears and we look at these spore formers, we find that they can either be a terminal spore, and this spore, if you notice, is within the cell. The subterminal, not quite in the center, but not quite at the end, and then central, in the middle of the cell. What we see happens is a vegetative cell, as you see here, gets some form of a signal that says things are going bad, things are running out. Starts to develop an internal structure, laying down different layers of proteins and things like that that will surround a copy of the genome, some of the enzymes. Not a whole lot, but some. And then you get this mature spore. The cell itself, what was the vegetative cell, that dies, either releasing the spore, or the spore can still be encased in the dead husk. Well, at some point in time, water comes back, nutrients come back, whatever that stimulus that caused this thing to change, well, that goes away. This spore will split open, and out comes a new vegetative cell to start this cycle all over again. The making of the spore, they refer to this as sporulation. Okay? The vegetative cell starts differentiating, making copies of stuff inside of itself. Not so it can divide, but so that it can pack these things away, almost in a panic room. It's an analogy I like to use when I teach this to, in other micro courses, especially for the nursing micro, the micro for health sciences. I'm trying to give this a means for them to understand it. Something bad has happened, something bad is changing, so the vegetative cell is going to put copies of stuff, copies of polymerases, copy of chromosomes, copies of ribosomes into this panic room. where it can remain metabolically inactive for days, months, weeks, years. Nutrients come back, water comes back. Germination will occur. This germination is that spore splitting open, releasing a new vegetative cell. So when we look at this, we look at these bacteria. Not all microbes can do this. Not all bacteria can do this. Only a select few have the genes with the instructions on how to do this. Well, we can utilize this as one of the key things we can use to determine bacterial genus, bacterial species. There are special stains to look for the presence of these endospores. We now know that the spore itself is actually a very complex thing. There are multiple layers, a core, an inner membrane, a cortex, outer membrane, a spore coat, and an exosporium. Each of these layers is going to have a completely different composition. With the different compositions comes different roles in, the heat resistance, the UV radiation resistance, the drying resistance, and all of that. The core at the center, where the chromosomes and the different enzymes could be, are going to contain what are called SASPs, small acid-soluble spore proteins. These will bind and protect the DNA. And when it's time for that spore to open up and release a vegetative cell during germination, these will become the carbon energy source for that new vegetative cell. So when we go through and we look at these endospores, you can see some of them are going to be free spores. Others are going to be contained within that dead husk of the original cell. When we go and we look at these differences between the endospores and the vegetative cell, calcium content. Turns out calcium is going to help stabilize many of these different coat layers. Because remember, each layer, different composition. Going to use calcium. Going to use diplochonic acid as a means to stabilizing, to aid in the resistance. Enzymatic activity, respiration, macromolecule synthesis. While it's going to be present and highly active in a vegetative cell, it's going to be low or completely absent in an endospore. That's why I keep mentioning the fact that they are metabolically inactive. When you compare the two, it's night and day. The water content. Vegetative cell, 80–90%. Endospore, 10 to 25% in the core. This is getting down to basically beef jerky, if not less. There's more water content probably in the piece of paper you have sitting on your desk right now than there's going to be in the core of some of these endospores. So when you go and you look, you'll see it even shows up on an electron microscope picture that the coloration for the different layers is different because their composition is different. And it shows up the core, inner membrane, a cortex, an outer spore membrane, okay? Please do not mix this outer membrane, this outer spore membrane. Do not mix this up with the outer membrane of a gram-negative cell. Two completely different things that have two completely different roles. The sporulation begins with, an asymmetric cell division. Okay? Normally, when most of these bacteria go through and divide, it's an equal division. Make a copy of everything, split everything down the center. Well, we see here it's asymmetric, and this new cell is going to be engulfed by what they call the mother cell. Okay, so instead of one long one being split in half and now you have two separate, you have this one, and the other one's going to be encapsulated. It's going to be engulfed. It's going to be within the other. We see a lot of the spore-forming species, like 200 plus of them, within Bacillus genus. Turns out that if you look just at Bacillus subtilis as an example, there are over 200 genes that are responsible for the making and the maintaining of a spore, as well as how that germination will occur. So when you look at it, you have a vegetative cell that's happily doing its thing. It's metabolically active until, and here it's the vegetative cycle. It just keeps growing, making new. Until a signal or a lack of a signal, that's basically triggers those 200 plus genes. You now get that asymmetric division, and that new smaller cell is engulfed, encapsulated by the larger mother cell. So instead of splitting here and you now have two separate, this one stays attached, now becomes engulfed. And the mother cell will begin laying down layers upon layers. Outer membrane, cortex, inner membrane. All of those different things get laid down more and more and more until the spore is finalized and that mother cell will die. As I've said in previous slides, that spore can either be released and be a free spore, or it'll stay within the husk of the dead mother cell. Either way, that mother cell is